So I'm Vanita Noronha, and I'm not the editor-in-chief of South Asian Journal of Cancer. That was maybe 5 to 7 years ago. So I'm currently editor-in-chief of Cancer Research Statistics and Treatment, a journal. I'm going to talk about phase 2 trials, the critical interpretation and assessment. These are my contact details, so feel free to reach out to me if any questions come to you after the session. So this is my Twitter handle. This is the Twitter handle of our journal. This is my email ID. So these are my disclosures. And just briefly going over the types of clinical trials, and I know all of you know all of this. We are going somewhat in backward order. So we started with phase 3 and now we're doing phase 2. So phase 1 are usually first in human trials. They can enroll healthy participants, very small sample sizes, and are used to really investigate the toxicity of a drug and the PK and PD. Phase 2, which I will be focusing on in this talk, are typically a smaller sample size, and they are conducted to get a main idea of safety, so of toxicity and some hint of efficacy. And phase 3 are generally the large trials. You have an investigational arm, and then you compare that to your standard of care. Phase 4 are post, after you have established the efficacy. It's when it's used. It's more of a real world setting, and they are large, and they're usually to document for marketing purposes done by pharma studies. So what is the idea of a phase 2 trial? So once you've completed your phase 1 trial, you know that the drug is safe, and you have established the dose that you want to use the drug in, and then you want to take it to phase 2. And is what you have discovered, your new drug or your new intervention, is that something that will eventually challenge the standard of care? So the idea in completing your phase 2 trial is to see whether you can take your intervention or your drug forward to phase 3 or not. And if your phase 2 study is quote unquote successful, so if that's a yes, then there's a benefit to your drug or your intervention, and the next step would be to take it to the phase 3. Sometimes you get a phase 2, which is not a positive study, but you see a signal in some conditions. So for example, in a subpopulation of patients, and then you may want to take it to phase 3 in only that subpopulation of patients. And if your phase 2 is negative, and that's usually the majority of phase 2, unfortunately. So that means that drug or that intervention is not efficacious, and then you don't take it forward. So the most important is to define your question, and you need to identify. So how do you identify a question for your phase 2 study? So you need to identify an area of unmet need. Sanchita, is there a way that I can get the participants or the attendees to interact? So maybe unmute and answer me some sort of situations that an unmet need could be identified in the clinic.
Yeah, so ma'am, they can put in the chat box as well, as in when you put in the questions, or they can raise their hand and the Riverroot team will actually unmute them. So either of it, we can do it.
Okay, so
Rohan, if you can help.
Yeah, so if any of the attendees or the participants could either raise their hands and tell me an area of unmet need, a possible area of unmet need, or put it in the chat box. So I'll start to kind of help you along the thought process. So one area of need could be, for example, in patients with locally advanced head and neck cancer who are non-metastatic and who would be potentially eligible for chemoradiotherapy. So in the most trials, patients who are over the age of 70 years have been excluded. So most of the inclusion criteria end at age 70 years. And in the MAC-NC analysis as well, there is no signal of benefit of chemoradiotherapy. So a possible question for us could be, in patients who are over the age of 70, does the addition of chemotherapy, concurrent chemotherapy to radiotherapy improve outcomes? Ajay Krishna has suggested utility of brachytherapy in early stage lung cancer. So absolutely correct. So could you, Ajay, if you could unmute yourself, can you tell me what your question is here?
Ma'am, hello ma'am, can you hear me ma'am?
Yes, go ahead.
Ma'am, the unmet need in this case would be like in early stage, like T1, T2, N0, surgery would be the preferred modality. So like for a radiotherapist, the unmet need or the question in this case for probably a phase 2 trial or a phase 1 trial would be whether brachytherapy can adequately cover a small disease which is limited to the lung, like a T1, T2 lesion in the lung.
Absolutely right. So eventually your question will be that can brachytherapy in this early stage lung cancer be an alternative to surgery? Is that correct?
Yes, ma'am.
Absolutely, absolutely. So that is one area of unmet need. Excellent. Any other suggestions on how you could identify an area of unmet need? And we see this every day in our clinic. Anybody else? I have just returned from ASCO and I attended a session on teaching on Zoom and how to interact with the audience. So I'm not going to move on until at least 2 or 3 of you answer questions. Okay, cool. So I think I will move on. So there are multiple. So, let me go on. So poor outcomes. So that we have situations in which there are very high rate of adverse events. So for example, the weekly, 3-weekly trial. So when we, the standard landmark studies have told us that using high dose cisplatin at 100 milligrams per meter squared is improves outcomes when you add it to concurrently to radiotherapy in locally advanced head and neck cancer. But extremely high rate of adverse events, 70 to 80% of patients in the trial had grade 3, grade 4 adverse events. So that's an area of unmet need. Could you lower the dose? Could you change the schedule? Poor quality of life, costly or inaccessible. So multiple standards of care globally are not applicable to our population. So for example, immunotherapy, the plenary session at ASCO on trastuzumab deruxtecan, not available to our population. So in patients who have HER2 low tumors in our setting, is there something else that we could use? Uncomfortable, so IV infusion, so full FOX, full FIRI, continuous infusion 5-FU. Could we substitute capecitabine? Could you give a drug orally? Could you give it subcutaneously? Resource consuming, so things that require a lot of manpower, money, time. So in our clinic every day, there are multiple areas of unmet need, especially in our setting, low middle income countries. So this would be, each of you could perhaps choose one area and work on that and try and answer that question. So let's move on. So it's important to remember that a phase 2 trial is not a confirmatory trial. It provides you an indicator. It gives you a hint of efficacy. And usually the type 1 error is large. So it's typically set at 10 to 20%. And just to remind you about the type 1 and the type 2 errors, I don't know, have you all gone over that? Athanu, has that been covered in earlier sessions? So this is the story. All of you know of the story of the boy who cried wolf, right? So this is a famous. Okay, so Dr. Krishnan has told me it has been covered. So this is the story of the boy who cried wolf, right? So he went. This boy was in a village. He was minding the sheep, and he thought, hey, let's tease the villagers. And he called wolf. There was no wolf. Initially the villagers came running, and there was no wolf. And he laughed and said, hey, I was fooling you. And he did this a couple of times, and then the third or the fourth time he called wolf, and the villager said, he's just teasing us. And actually there was a wolf. So this first setting, can anyone tell me, is that a type 1 or a type 2 error? So in the first setting, so in order to understand if it's a type 1 or a type 2 error, you need to say what is the null hypothesis here. So in the first case, when he cried wolf, but there actually was no wolf, what is the null hypothesis? And maybe you can just put it in the chat. Or so first setting, type 1 or type 2, and second situation, type 1 or type 2. So in the first situation, the null hypothesis is that actually there is no wolf, right? That's your null hypothesis. There is no wolf. And if the visitors did come, so they rejected the null hypothesis. So rejecting the null hypothesis, so that's a false positive. That's your type 1 error. In the second situation, again, the null hypothesis was that there was no wolf, and the villagers accepted that null hypothesis. So that is your type 2 error, right? Is that clear to everybody? Absolutely correct. So first setting type 1 and second setting type 2. So in a phase 2, we set the type 1 error high. So you accept that there is a higher chance of a false positive because you want to take the drug and test it further. You don't want your chance of saying that the drug is effective. You want to develop as many drugs as possible. So you set your type 1 error higher. Is that clear? Okay. So let's go over a few of the designs of a phase 2 study. So phase 2 study could be a single arm study or a randomized phase 2. The randomized ones nowadays are far more common, and it could be a non-comparative randomized or a comparative randomized. So a non-comparative randomized study, you have a reference arm, and so you're not randomizing the patients. So you have a reference arm, and I apologize. So in the non-comparative, you have a reference arm. You're not comparing the two arms, but the reference arm is just to tell you that in your set of patients. So for example, if a drug has been developed in another setting or globally, but not been studied in India, you want to see if your outcomes in the general in that setting are different than what have been actually described as a standard of care. So you keep a reference arm, but you're not really comparing the two. In your comparative randomized, you can have multiple arms, and in which you set your predefined hypothesis and you pick the best arm, and that is the arm that goes on to the phase 3. You can have a randomized discontinuation comparative study. So both arms will have the drug or the intervention, and then after a certain period of time, or you define the response, and then after that, in one arm you discontinue it, and in the other arm you continue that intervention, or you can have a randomized phase 2. And again, that is the most common current design, the randomized phase 2, and that could be open label or blinded. So what are the implications of choosing your design on the results? So the randomized phase 2 is most common, and that has the highest level of interpretation and the highest, best evidence generated, but it is resource consuming, and it's perhaps sometimes unrealistic if the situation that you're studying is rare or has a very poor prognosis. It could be non-comparative, and that's the next best. So there is an arm that is non-randomized, and as I mentioned before, so that's just an idea to see if your population, the outcomes are worse than what has been earlier described. And then you can have a single arm phase 2, which is not done as commonly. That could be a 2-stage or a 1-stage design. So just to give you a couple of examples, this is a study done in head and neck cancer. So head and neck squamous cell carcinoma, patients with ECOG performance status of 0 to 2, requiring palliative chemotherapy and adequate organ function. So they are randomized, and this is the first stage. So this is the phase 2 part of the study. So they are randomized 1:1:1. So arm A is paclicarbo, and 44 patients would be enrolled on that arm. Arm B is paclicarbo with triple metronomic, so celecoxib, erlotinib, and methotrexate. And arm C is paclicarbo followed by celecoxib, erlotinib, and methotrexate. And the primary endpoint is PFS in the phase 2. At the end of the phase 2, these two arms, so these two arms are not being compared to the standard arm. So you just have a standard arm as your reference. And these two arms, based on the PFS, there's a futility analysis written into the study. You discontinue the futile arms and pick the winner. So the one that had the PFS that you had predefined, and that goes on to the phase 3 part. So in the phase 3 part, 163 patients would be added to each of those arms and the arms that you chose. And then that would be compared to the paclicarbo. So and in the phase 3, the primary endpoint would be overall survival. So this is an example of an open label multi-arm. So open label, the patients as well as the investigators know what kind of treatment is being given to in each arm. Multi-arm, so you have multi-arms. It's multi-stage, so it's phase 2 and a phase 3, and superiority. So in the phase 3, you're looking for superiority. So is that clear? Okay, so this is another example of a multi-stage design. So this is patients with recurrent glioblastoma. So would be, and this has multiple iterations. So if they are eligible for re-irradiation, if yes, then they get placed on arm A, which is radiotherapy plus temozolomide plus mebendazole. If they are not eligible for re-irradiation, then they get randomly assigned to arm B, which is CCNU plus mebendazole, or to temozolomide plus mebendazole. Now, this is actually a phase 1. So this is the first is the phase 1. And so there's a dose escalation to find the maximum tolerated dose. Going to the phase 2, and in the phase 2, that is the that's the second stage, and it's phase 2. And in the phase 2, the endpoint is a 9-month overall survival. One stage, so talking a little bit about one-stage design. So one-stage designs, the way to calculate the sample size for one-stage, single-stage phase 2 designs was described by Ahern, and this is a program in your statistical software. And this is a program in R. It's phase 2 single, and it gives you on the top. It will give you these values. So PU is the unacceptable response rate. So you say if the response rate is lower than PU, I'm going to reject my hypothesis. I'm going to reject this this drug or this intervention. PA is your acceptable response rate. So that's the response rate that you hope for. And then if you attain that response rate, you go on to the phase 3. EP1 is your threshold for the probability of declaring the drug desirable under the null hypothesis. So can anyone tell me what is EP1? Is that your type 1 or your type 2 error? So none of you listen to the story of the boy who cried wolf. So EP1 is your type 1 error. EP2 is your type 2 error. So that's the threshold for the probability of rejecting the drug under the alternative hypothesis. And N solin is the number of designs with the given alpha and beta. So this is one example in which this program was run. So it gives you 5 possibilities. So you set your type 1 and your type 2 error, and that's a clinical judgment. You set that. N is the sample size, and R is your decision point. So in the first where you set your type 1 error of 4% and your type 2 error of 4.7%, your sample size is 98. And if 26 patients respond, then that is appropriate and you move on to the next level. And if you remember the first one, the 0.2 is your lowest response. So if your response is 20% or lower, then you reject it. If your response is 35% or higher, then it is positive. This is your type 1 error 5%, your type 2 error 5%, and N solin is the number of the program just gives you 5 options. So this is one example again of a phase 2 study. And this is and we had calculated the sample size from using that Ahern one-stage design. Again, most of these designs have been, I've taken them from Dr. Vijay Patil. He has calculated the sample sizes and has been kind enough to share his slides. So this is in patients with recurrent glioma, 46 patients, all enrolled on, treated with low-dose bevacizumab at 1 milligram per kilogram plus CCNU. So patients who are enrolled have recurrent glioma, performance rate is 0 to 2, no uncontrolled comorbidity, and not feasible for local treatment. And the decision rule here is that 15 or more patients, so 46 patients were enrolled, and the outcome is overall survival. So the decision tree is that if 15 or more patients are alive at 6 months, then the arm is taken forward. So coming to the 2-stage, so is the 1-stage clear? Any questions at all? There's something in the chat. Phase 1 does not have a comparator arm, does it? No, phase 1 does not have. Yes, type 1. I think that you were answering the previous question. So yes, that is type 1. Ajay, I think you're the only one who's actually listening. Huh? Okay, so going on to the 2-stage. So the 2-stage design, so in the first stage, you enroll a certain number of patients. If X1 or more patients respond, you go to the stage 2, and then you enroll an additional N2 number of patients. If less than that number that you have set respond, then you stop the trial. So it has been, the sample size calculation has been described by Simon. And again, this is the software to be used in the calculator. So the phase 2 Simon sample size calculation, the PU again is unacceptable response rate. So if your drug leads to that response rate or lower, then you reject that drug as being efficacious. If it is the PA is the number that is the response rate that you want, and if you attain that response rate, you go forward. EP1 is your type 1 error, EP2 is your type 2 error, N max is the it's default of 100, so that's your total sample size. So it defaults to 100, and you can increase that and say, and you can decide that based on whether you decide on an optimal or a minimax design. It's somewhat out of the scope of this talk, but perhaps Dr. Athanu can talk to us a little bit about that later. So this is an example. So this is from R. So a Simon two-stage phase 2 design. So the unacceptable response, so you're choosing a drug X. If the response rate is lower than 20%, you reject that drug as being effective. If your response rate is 35% in your phase 2, then you say that it's acceptable and I will move on to phase 3. Alpha error of 0.05, beta error of 0.05. In the first, so R1 is your first stage, N1 is the sample size of your first stage. So in a sample of 50, if 11 patients respond, you move on to the second part of the phase 2, and you go on to enroll 109 patients. And if 28 of those 109 patients respond, then you say that I'm going to take my trial forward. Using the minimax design, you set the minimum number. So the minimum number of patients that you will accept as, saying that this is possible for me. And so choosing minimax design will give you the lowest sample size possible. And optimal is a little bit higher than that. And I would recommend that you go back and read this paper to have a little bit better understanding of this optimal and minimax and admissible two-stage design for phase 2 oncology trials. So just an example now of a phase 2 study with biomarker analysis. This was just published. This was using afatinib and pembrolizumab in patients with recurrent or metastatic head and neck squamous cell carcinoma. This was the alpha study. So this is a phase 2 single arm trial. The primary endpoint was the objective response rate, and it was based on Simon's two-stage design. So the null hypothesis was that the true response rate is 15%, and that would be tested against a one-sided alternative. So we wanted, 15% was your, I'll just go back, 15% is your PA, right? So that's your response rate that's desirable. So in the first stage, 13 patients were recruited, and so 15% of 13 would be 2. So if 2 or fewer responses were observed in these 13 patients, I think I made an error. So 15% is actually 15% is your stopping rule. So if 15% or less patients respond, then you reject it. So 15% is actually not your PA, it's your PU. So less than 15% respond, you reject it. So if 2 or less out of 13 patients respond, then the trial would be stopped. And if more than 13 patients responded, then you moved on to the second stage, and then you would recruit 16 more patients to a total of 29 patients. And in those 29 patients, if 8 or more responses were observed, then you would say that this study is, 8 or more responses were observed, the null hypothesis would be rejected. The type 1 error was 0.05. The power was 90% for the true response rate of 40%. Is that clear? At any point, if anyone has a question, just please raise your hand and feel free to interrupt. So going on to the next type of phase 2. So you can have a seamless phase 2 to phase 3 design, and that's perhaps the best way to do a study in our setup. So if you didn't have a phase 2 going through to phase 3 design, the problem is that the patients whom you have enrolled on your phase 2 design, they will not contribute to the phase 3. So it's a lot of, you'll have to again do that, the whole process again, write a phase 3, submit it to the ethics committee, get it approved, and then you'll have the entire sample size to recruit again. On the other hand, if you had a phase 2 going through to phase 3 design, the patients in your phase 2 will contribute to your overall analysis of the phase 3. So it's a good utilization of resources, but it is a bit complex. And there is, in certain situations, you can have sample size re-estimation. So that's an uncommon situation. And you have to go back to your ethics committee in between, and if something has changed in terms of the standard of care, or if you see that that there may be a benefit, but you're very borderline, then you can re-estimate your sample size. So I see a question in the chat. Stage 1, phase 2. I'll take that at the end of the talk, Ajay. So I just wanted to talk about one of the study that I had just published. So this was a study in which we evaluated the role of oral metronomic chemotherapy after definitive chemoradiotherapy in esophageal squamous cell carcinoma. So this was a phase 2 going to a phase 3 study. So we included patients with squamous cell carcinoma of the esophagus or GE junction who had completed definitive radical chemoradiotherapy, were adults, had an ECOG performance status 0 to 2 and normal organ functions, and excluded patients who had clinical or radiologic evidence of metastatic disease or progressive disease. So after they completed chemoradiotherapy and after 2 to 3 months, they underwent restaging. And if they did not have any evidence of disease or PD, they were then randomized to observation, which is the standard arm, or to oral metronomic therapy for a year. The primary endpoint of the phase 2 study was PFS, and of the phase 3 study was overall survival. So this is what we had detailed in our protocol as well as in the final paper. So the primary endpoint of the phase 2 was progression-free survival, defined as time from randomization to disease recurrence or death in the absence of disease recurrence. So this goes back to, I think, one of the questions in the previous talk to Dr. Kumar. So what do you include as events for disease-free survival or progression-free survival? And that's, again, it's very important for you to predefine that. And this we had predefined in our protocol, as well as we required to define it in the paper, because this is a bit somewhat unusual. So for us, events for progression-free survival included disease recurrence, identified radiologically or clinically as clinical deterioration or death. Now, clinical deterioration is not commonly included as an event for progression-free survival. But I think over the years, we have realized that in our center, at least, patients go back to local place, especially after they complete their primary therapy. And then when at local place, many times they don't have access to a doctor. So I had written this, and this was accepted by the ethics committee as well as by the editors of the journal. So if patients develop disease-related symptoms or clinical deterioration at home, they many times cannot travel back to our center. They don't go to the doctor. So we don't have radiological evidence that they have progressive disease. But we thought, let's be conservative and use a worst case scenario. And so we coded all of these as progression events, and they contributed to the progression-free survival. In the phase 3 portion, the primary endpoint was overall survival. And so to calculate the sample size, it's important for you to have your own data or to go back to the literature and see what has been described in the literature earlier. So we had earlier published our retrospective data of patients with locally advanced esophagus or GE junction cancer who were treated with radical chemoradiotherapy. In those patients, the median progression-free survival was 11 months from the start of chemoradiotherapy. Now, in our study, we would randomize the patients about 3 to 4 months after start of chemoradiotherapy. So after they completed chemoradiotherapy and then underwent staging. So we set our baseline assumption for PFS in observation arm of 8 months. We set the power of 70% because this was the phase 2 portion, alpha of 10%, and we hypothesized that a hazard ratio of 1.5. So the p-value for stopping the trial after phase 2 was 0.2 for the progression-free survival. And then for the phase 3 portion, we assumed the median OS would be 14 months, which we extrapolated from global literature. Power of 80%, one-sided alpha of 0.025. So we hypothesized a hazard ratio of 1.43 and required 151 patients. So 151 patients were enrolled in phase 2. And then for the phase 3 portion was 262. But this 262 would include the 151 patients. So this was an optimal phase 2 going to phase 3 design. So after phase 2, we analyzed the data and we found that this was not a positive trial. So the blue line actually represents the interventional arm. So the patients who received oral metronomic chemotherapy actually did worse than patients who underwent observation. So we stopped that. We stopped it at phase 2 and did not go to phase 3. So this is something for you when you are evaluating a phase 2 study. You need to look at the methodology. Was an appropriate design used? Was the population appropriate? Was an appropriate control used? And what was the power calculation? Now, this was actually supposed to be the next talk I did. So this was actually now the previous talk that Dr. Kumar has already discussed, how that the population in the study has to reflect the population of patients that you're going to be applying the results of the trial to. So the patients whom you're seeing in the clinic, was there an appropriate active control used? And was it appropriately powered to answer the hypothesis? Well, is the methodology detailed so that you can reproduce what the investigators have done in your setup? What about endpoints for phase 2 trial? So key endpoints, so phase 2 is normally quick. So you want to know whether you want to go to the phase 3, which is a long trial. So it should be a good surrogate endpoint, and it is usually a response rate, progression-free survival, or disease control rate. But that endpoint has to be appropriately described. So you have to define what events will contribute to deciding on progression-free survival, which patients are you going to censor, and when are you going to assess your response? Is it at 6 months? Is it a median? So that needs to be estimated. So randomized phase 2, you compare between multiple arms for your primary endpoint, but only that can be done if you're powered appropriately for those comparisons. And you can compare each arm against your historical control if it is a pick the winner or you're stopping any arm. Again, non-randomized, the decision rule. So if X number of patients respond or have not progressed at a Y time point, that arm will be considered for further evaluation. And but you need to make sure that you've framed your decision rules at the time of sample size calculation. So I think in the interest of time, I'll skip this, but this is a very important paper for all of you to go back and read. So this is how to report phase 2 clinical trials. And this was a paper published in Annals of Oncology about 7 years ago. And they reported on quality of phase 2 trials. So the publication. And they took the trials that have been reported in high impact journals. So about 37% of the trials were from JCO and 25% were from Annals of Oncology. And these are all the criteria that need to be present in your paper. So the title has to indicate that it's a phase 2, whether it's single arm, randomized, and what type of intervention. The introduction has to have your study rationale. The methodology has to be detailed, your primary objectives, your secondary objectives, the type of patients that you're selecting. So describing the population that have been enrolled in the trial, describing the treatment that is being studied, the dosing, the number of cycles, what constitutes a completion of the trial, what are your dose modifications, what are concomitant medications, an adequate description of your study procedure, so imaging and the type and the frequency of imaging. Was there a central radiologic review? When were you doing labs? And the statistical methodology. So sample size calculation, which was your evaluable population? So which population went for efficacy analysis? Which was your safety population? What statistical methods did you perform? Did you use an intention to treat analysis or a per protocol analysis? And your results. So the dates of inclusion, the setting, which patient, the number of patients who were included, number who were excluded, ineligible, what was your median follow-up, what were the reasons that the patients discontinued treatment, the characteristics of the patients, flow charts, the analysis, so number of patients that you included in each analysis, your confidence intervals, and safety. And in your discussion, your conclusion has to be the primary endpoint consistent with the results provided and with your initial hypothesis. So they scored the quality of phase 2 trial reporting based on all of these criteria. And you can see the overall quality score. The maximum score would be 44, and the median score that these phase 2 trials published in high impact journals was only 28. So quite poor. And they made something called a key methodological score. So these are the 3 things that must be there when you are planning and reporting your trial. So the primary endpoint, the sample size, and describing your evaluable population. And only 67% describe the primary endpoint, 77% describe the sample size calculation, and only a third describe the evaluable population. So these are really important things to keep in mind. So I'll end here. It's important to decide whether to do. So a phase 2 trial is important to do to help you decide whether to do a phase 3 trial or not. And the things to look out for that may bias this decision. So selection bias, a weak control arm, an inappropriate design, inappropriate endpoints, and inappropriate interpretation. So I'd just like to acknowledge Dr. Kumar Prabhash, who was the first speaker, and Dr. Vijay Patil. So both of them have taught me. I don't have any background in statistics at all or clinical trial methodology. So I think whatever I've learned from the two of them. and Vijay actually made and helped me to make these slides. So thank you very much.